Welcome to Lab 7. So in this lab, we are going to be covering bioarchaeology and forensic anthropology. So before we get started, this is just a note, like we've covered the bones, we're familiar with the bones, and now we're going to take our knowledge of the bones and then apply it to the actual field of anthropology. So when we think of bioarchaeology and forensic anthropology, we know that in general, coroners and other related experts, they have a valuable understanding of the human soft tissue. However, a lot of times there are situations where the soft tissue evidence is limited or it's completely unavailable. In these situations, an understanding of the nuances of the human skeleton can be very, very useful. And so we'll see with both bioarchaeology and forensic anthropology how we can take our understanding of the human skeletal system and then apply that to understanding different scenarios. So let's go ahead and get started. And the first thing we're going to do is we're going to answer, what is bioarchaeology? So bioarchaeology is the study of human skeletal remains from archaeological sites. So one key thing here is with bioarchaeology is that we're not involved in contemporary legal cases. We're more interested in understanding the past lifeways of historic and prehistoric people through the analysis of their skeletons. So bioarchaeology studies attempt to reconstruct past human activities, their diet, their disease, their overall health patterns. So because bioarchaeologists apply the methods of skeletal analysis to archaeological contexts, they then bridge the gap between biological anthropology and archaeology. Like many other fields, bioarchaeology is often interdisciplinary and will overlap with other disciplines such as demography, chemistry, and medicine. How much information that bioarchaeologists can learn from skeletal remains really depends on the state of preservation. If you have a really well-preserved site, then you're going to get a lot of information. If the site has been damaged or people have gone in and taken things, or it's just not conducive to preservation, then you're going to have like a limited amount of things that you can learn from that site. So preservation is affected by multiple factors, including how old the remains are. So the older they are, probably the less well preserved they're going to be. And in what type of environment they're buried, right? Is it dry and arid? So it might be more conducive to fossilization. Is it moist? Is it humid? That means rapid decay. Rapid decay is not conducive to fossilization. So those kinds of things can affect how much information we are able to obtain from a site or from skeletal remains. Additionally, the study of ancient skeletal remains carries important ethical and legal issues. So the one thing with anthropology and in general is that we have to keep in mind that there are ethical issues that might arise at any given point. And so we always have to be in awareness of what is the ethical way of conducting our research. And there's a lot of different ways we can approach this. And like in the exercise in the first week where we asked about ethical issues, there are a lot of different ways to approach ethical issues. And so that's one thing to keep in mind. So with bioarchaeology, we might run into ethical problems or ethical dilemmas. One example I can give is that in many countries, including the United States, there are laws that protect the rights of descendant communities and their religious beliefs and cultural heritage over the study and disturbance of skeletal remains. So there's been a lot of cases where say skeletal remains are discovered for whatever reason, and then they don't know who they belong to. And then it emerges later on that they do belong to a descendant population or a community, like a native indigenous population. And so in that case, what happens is that those skeletal remains usually are then returned to their descendants, and then they can bury them or dispose of them in a way that is meaningful and in alliance with their religious spiritual beliefs, right? There's two examples that I think of. One was more recent. It happened, I believe it was here in Southern California. So if you're familiar with the 405 freeway and the ever continuing expansion of that freeway, there was a spot on that freeway where they did discover human skeletal remains. And so it was over by around the Huntington Beach area. And so they had to stop doing construction for a period of time because they had to go in and they had to investigate if this is a site that there was more skeletal remains there. Was this because it was indigenous land, or was it something else? So they had to go in and, you probably got both bioarchaeologists and forensic anthropologists going in and then studying and then figuring out. I think eventually it did come up that they were native indigenous skeletal remains, and they were then returned to their original descendants, and then they took over from there. In a lot of cases, that might mean that construction has to stop, right? And so I'm not sure what happened after that in that case, because I never followed up with it, but I do know of another example that happened in Florida. Kind of the same situation. They were like demolishing this like parking lot, I believe, or area, in order to build like a little mini strip mall or something like that, and they found like an archaeological site. I don't think they found skeletal remains, but they found some kind of archaeological site, and they ended up bringing in anthropologists and archaeologists to come in and investigate. They investigated. They found out that this was in fact a important landmark, and so they actually had to stop 100% all construction on the site, and they could not build on top of it anymore. And then the last example that I think of, I know I said two examples, but I just thought of that second one while I was talking about the first one. Another example is the example of the Kenwick Man. And the Kenwick Man was a set of skeletal remains that I had heard about when I was in undergrad actually, right around the time where I was taking physical anthropology. So I remember hearing about it. That was around the time it was discovered, or it was a new discovery at the time. And they call it the Kenwick Man because it was found near Kenwick, Washington. And it was a set of skeletal remains. And they had no idea who it belonged to. And so they studied it. They were analyzing it. Finally, and so that was, around when I was in undergrad. So very, very long time ago. And so, let's see. It was like 2008. 2000, let's say 2009, 10 is around when I took physical anthro for the first time. So it was around there, and it wasn't until maybe a couple years ago, maybe two years ago, so 2019, when they finally 100% had determined that the Kenwick Man did belong to an indigenous population of the region, and so they then returned finally those skeletal remains to the indigenous tribe that he belonged to, and then they took care of it. So again, there are laws that do protect the rights of descendant communities and their religious beliefs and cultural heritage. NAGPRA is a very important organization. It's the Native American Graves Repatriation Act or something like that. Can't remember off the top of my head. They are in charge of, again, ensuring that those remains do stay either where they're at, or they're undisturbed, or they're given back to their descendants, essentially. And they do other stuff too. Those are just some examples. As bioarchaeologists, we compile as much information as possible from ancient skeletal remains to reconstruct the life of an individual. So their age at death, their sex, their signs of disease, anything that you can get from the skeletal remains, that's what we're looking for. Ultimately, we're also interested in reconstructing the lives of groups of people and the communities, not just the individuals. So we're going to get a skeletal remains set, but we're really, really interested. Well, we're interested in this remains, right? We want to know everything about them. But what we're also really interested in is everything about the group of the people that it lived with, right? Everything that we can reconstruct from these remains. So we apply many of the same methods and techniques as forensic anthropologists, and when presented with remains, we may follow many of the same protocol steps. So we will do the same thing. You're going to see that the steps that we cover, they're the same for bioarchaeology and forensic anthro, except forensic anthro has a few extra steps at the end. Bioarchaeologists study the life histories of archaeological populations to get a sense for how the larger group lived and to answer broader questions. Some of these questions are things like, were there any differences between the way males and females lived? Or differences between the way the young and the old lived, or different statuses, right? We can often figure out these things by looking at the skeletal remains, right? So that's bioarchaeology. Now, what is forensic anthropology? I know I've talked about it a little bit, but what really is it? So forensic anthropology is the branch of biological anthropology that applies specialized methods of skeletal analysis to human skeletal remains during the course of legal investigation. So an example would be if a victim's body was buried for many years or was severely burned, right? There's a lot of the victim's soft tissue no longer available. And because it's no longer available, a medical examiner or coroner can't really, not that they can't, they might have a hard time identifying cause of death, if there's anything suspicious about the death, anything like that. So when a lot of the soft tissue is no longer available for then a forensic anthropologist is called to analyze the remaining skeletal material. So the primary job in this situation is to build a profile that will help then in identifying the victim and, if possible, help in determining the circumstances surrounding the victim's death, right? So the job is to build a profile. That's the job. It's not, this is the cause of death and this is how it happened, right? Popular TV and shows and books will suggest that forensic anthropologists spend all their time investigating individual victims. That's not really the case. We're really just called onto these cases as they happen, right? So essentially we build that profile. So again, it's not always the case. We're not always investigating things. Forensic anthropologists often work on cases of mass death, such as genocide, war crimes, and natural disaster. But many forensic anthropologists really participate in investigations part-time. So it's not like we do this 100% of the time. A lot of forensic anthropologists really spend the rest of their time teaching or conducting research. So when forensic anthropologists are investigating skeletal material in legal context, they follow a protocol that lays out steps in a meaningful order to help them identify an individual. Often, one step cannot be accurately completed without the completion of prior steps. So you have to do step 1, then step 2, then step 3, 4, 5, 6, 7, 8, right? You have to go in order, right? So the steps and their order are going to be very important, right? It's highly likely, and I say this with a guarantee that there is going to be a question where I ask you, like, okay, what comes before this step, or what's the first step you're supposed to do on our quiz? So make sure you know the steps. So again, they cannot accurately be completed without the completion of the prior steps. These steps include determining the age, sex, and stature of individuals, as well as examining indications of pathology before death and estimating the amount of time since death. So these are some things that we look at as forensic anthropologists. So like biological anthropologists, bioarchaeologists, and forensic anthropologists are interested in examining aspects of human variation, particularly aspects of skeletal variation, right? This is where we get a lot of information. So differences in age, biological sex, height, or response to disease are all aspects of human variation that we can examine in the skeleton. So let me give you an example. The skeletons of males and females, they can differ in the shape and the size. Similarly, bones grow larger and teeth erupt at different ages and at different times in male and female children. And the bones of older adults will degenerate in a regular fashion and appear different from the bones of younger adults. All of these are key factors then that we can use to determine sex, height, response to disease, human variation, anything like that we can use. These are all key factors we can use. So when we're doing research, and research in general, not just forensic and bioarchaeological, there are two primary ways to study variation, right, in any research. Qualitative methods and quantitative methods. So qualitative methods are methods where we observe the variation of the bones by visual inspection. So I think of quality, right? And then quantitative methods is where we actually measure the characteristics of the bones or the teeth. So I think numbers. Anything that has to do with numbers automatically goes into quantitative methods. Anything that has to do with, say, has this trait, doesn't have this trait, is maybe, anything descriptive, essentially, will go into qualitative methods. So just keep that in mind. So anthroposcopy is where we observe the variation of bones by visual inspection, right? That's what we're looking for in qualitative methods. So the observation of physical bodily characteristics by inspection as opposed to exact measurement, right? So exact measurement would be quantitative, anthroposcopy would be qualitative. So an example is when we look at the differences in the shape of the pelvis between males and females, or the presence of certain tooth shapes, such as the shovel-shaped incisors we see in some populations. These are qualitative observations, right? We'll be looking, say, okay, do they have shovel-shaped incisors? Yes or no? Then that's, again, qualitative. Anthropometry is the scientific study of the measurements and proportions of the human body. So we might measure variation in the length of an individual's femurs, which contributes to our variation in height, or we may measure the sizes of their molar teeth, which may vary between the sexes. Often different human species are Neanderthals, Homo sapiens, erectus, all of them, and even among modern human populations. So we'll see, again, the differences in these sizes. These are exact measurements, so that goes under quantitative data. And then we have osteometry. And osteometry is the study and measurement of the human or animal skeleton, especially in an anthropological context. So those are different ways that we look at skeletal biological variation. Both qualitative and quantitative approaches are used in bioarchaeology and forensic anthropology to learn more about the lives and the identities of individuals from their skeletal remains. So now we have the background. We have the understanding of what is bioarchaeological and forensic anthropology. How do we do it, right? How do we actually conduct bioarchaeology, forensic anthropology? And so there are methods, right? So methods used in the analysis. Many of the steps and methods we use are the same for forensic and archaeological contexts, like I said. I think like the first 6 are the same, and then the last 2 are specific to forensic. So once the basic steps in analysis are complete, additional methods are used to gain more specific and specialized information. So I'm going to show you the first 6, then the 2 for forensics, and then we're going to go through each one one by one. So here are the key steps in analyzing human skeletal remains. So this is the order you're going to want to memorize. You're going to want to remember. It really makes sense, right? You're narrowing down information with each step, right? If you were given an array of skeletal remains, just a bunch of bones, right? You're going to want to figure this out in each of these steps to really understand and narrow down your information. So the first thing is to distinguish between human bone and animal bone, right? Automatically, you're going to be getting rid of some bones, right? Because it's not that we're not interested in the animal bones, it's just that we're more interested in the human bones, right? So distinguish between humans versus animals. Once you've done that, you're going to determine the minimum number of individuals present, right? We're going to find out how many people minimum do we have here. Next, you determine the sex of each individual, right? How many females, how many males. Determine the age at death for each individual, right? How many juveniles, adults, and so on. Estimate the stature of each individual, so how tall were they? And then identify pathology or stress indicators of each individual, right? So is there anything, disease or bone breaks, remodeling, anything like that that might have been prior to death or might have been around time of death and caused death. So these first 6 are the same for bioarchaeology and forensic anthropology, and then we have 2 that are specific to forensic anthro. And so the 2 is determine the ancestry of each individual and then calculate the postmortem interval. So how long has it been since death? So let's go through each one by one. And the first step, again, is to distinguish between human versus animal bone. So often, human bones can be mistaken for animal bones, especially when the bones are fragmented and incomplete. So there are 2 considerations when determining if a bone is human. First is to consider the size, right? Is this bone that you're looking at, or fragment, appropriately sized for a human, right? So an example is cows are very large. They have large, robust bones. So it's clear even in bone fragments which one would be a cow bone versus a human bone. So this then can be used to differentiate that between the cow bones, right, from the more gracile or the more slender human bones. Another example is dog bones are relatively gracile, but they're smaller than human bones. So the smallness can be used to differentiate them from larger human bones, right? So that's the first step. Consider the size. The second step here is to consider, are there any unique human adaptations? So there are adaptations in the skeletal system that are uniquely human, right? Chins and foreheads, right? Humans have chins and foreheads. All other animals do not, right? So if a mandibular fragment is found with a chin, it must then be human, right? Similarly, if a tall vaulted frontal bone is found, it must be human. Humans also have a generally large brain case with a smaller face, a shorter snout, smaller canines in comparison with our other animals. Other animals have smaller brain cases, larger projecting snouts, and in the case of carnivores especially, they have larger saber-like canine teeth. So anything uniquely human, you put them in the human pile. Anything uniquely not human, in the other pile. And then we also have a series of specialized adaptations for bipedal locomotion that makes our skeleton unique as well. Things like our angled femurs, the position of the occipital, not occipital lobe, the foramen magnum, the, foramen magnum position, the mastoid process, that's the word I was forgetting. S-shaped vertebral column, bowl-shaped pelvis, angled femurs, arch in the longitudinal arch in the foot, non-divergent hallux of your big toe, and there's something else that I'm missing right now. But we have these adaptations for bipedal locomotion, so that would help determine if these bones are human or not. So there we go. So that's the first step. Determine determine what is human and what is animal. The second step is to determine the minimum number of individuals. So in most bioarchaeological contexts and in many of our forensic contexts, the recovered bones are from multiple individuals. So it's up to us, the bioarchaeologists or forensic anthropologists, to determine how many people are actually represented by the bones in the assemblage, right? So our collected materials. So because there are many bones in the human body that could be recovered, it is often impossible to determine exactly how many people are represented. Instead, biological anthropologists will focus on determining what we call the minimum number of individuals, or the MNI. And this is what we have, the minimum possibly represented by the bone assemblage. So this gets us closer to the number of individuals without overestimating the number. So there's 3 steps to this process, right? How do we determine a minimum number of individuals? We do it with 3 simple steps. They're pretty easy. So the first step, are there any bones that are unique in the human body, right? Again, looking at bones that are uniquely human, right? Because there's only one mandible in each human, if your assemblage has 2 mandibles, you must have at least 2 people, right? You could have more than 2 people because there could be people in this assemblage whose mandibles were removed or destroyed, but you definitely at least have 2, right? You have a big box of bones and they ask you what's the minimum number of individuals. You find 2 mandibles, 1 left femur, 1 right femur, let's say 2 right humeruses, right? You still have a minimum of 2, minimum of 2. So there's only 1 mandible in each human. If your assemblage has 2, you have at least 2 people, right? And then the second step here is, are there any bones that are from the same side of the body, right? So some bones of the body are not unique, right? But they are sighted. You have 1 left humerus, 1 right humerus, 1 left femur, 1 right femur, right? Same thing with your tibia fibulus. So some bones are not unique. They are sighted. So if your site has 2 right femurs, then you know you have at least 2 individuals. So say you have 1 mandible, but you have 2 right femurs and 1 left humerus, right? Still have a minimum of 2 individuals, right? Again, you could have more, but you know for sure, you can say with certainty that you have at least 2 individuals. So step 1, any unique bones unique to the human body. Step 2, are there any that are from the same side of the body? And then step 3 is, can you match any of the bones to the same individual? So the final step is trying to match the bones together. Bones that appear to join with each other, such as the right femur and the pelvis, can be articulated to see if they fit correctly. So if they do, then the femur and pelvis belong to the same person, not to two different people. So biological anthropologists will also determine if multiple bones might belong with each other based on their preservation, condition, age, sex, or pathology. So bones from the same body and skeletal area are often in a similar state of preservation and similar age and sex. So the bones may also show that the same disease or pathology if they are found in a similar part of the body or if the pathology is spread throughout the body. So you can have pathology really help you also determine if it's from the same body. Right? So after we determine that the bones are human and we estimate the number of individuals, the minimum number of individuals, one of the first things that bioarchaeologists or forensic anthropologists must determine is the biological sex of the individuals in question. And this is what we call sexing. So determining the sex from skeletal remains is based on sexual dimorphism. This is the variation between adult male and adult female bodies. In the case of sexual dimorphism in the skeleton, we are specifically looking at the differences in bone morphology, our size and our shape. You'll see that when we talk about our other primates when we get to primates, you will see then that there are other non-skeletal traits of sexual dimorphism that we see in our primates. Things like brightly colored fur around genitalia, the sagittal crest in our gorillas, things like that. But when we're talking about forensics and bioarchaeology, we're specifically looking at the differences in bone morphology. So sexing of skeletons is relative and it exists on a continuum. So an individual is more male or less male, or more female or less female. So it's really, again, on a continuum. Sex determination can only be done on adult skeletons. So before we get there, I want to just make this, the continuum part very clear, right? Again, a skeletal remains will be more female or less female, or more male and less male, depending on where they lie on the continuum. But what this means is, and I want to give you an example. If we have the skeletal remains, a really tall set of skeletal remains, and then we had a shorter set of skeletal remains, right? Before I tell you the profession of these individuals, without knowing, we would assume, based on sexual dimorphism, that the taller ones, because this naturally happens among humans, men or males tend to be taller than females. And so given that rule, or that, tendency, we would assume that this taller individual is male and the shorter individual is female. Right? It's a safe assumption to make. But, again, we want to also take another, that's not the only consideration we would take into account, right? We would see other things, right? But if I tell you that the tall individual, right, was a WNBA player, and the shorter individual was a horse jockey, right? Then we would know WNBA player, then they're female, right? They're just taller than the average female. And our horse jockey, right, he might be shorter. And so we would have assumed, based on height differences between the two, that he were the female one. He is actually the male one, because again, it's just his stature, right? And I'm not saying the job is what gave him a smaller stature, or the female a larger stature. It's just that's just extra information, right? I'm just showing you that, again, it goes on a continuum. So it's not 100% there. There are other ways to get it, though. Like, height is not 100%. There are other ways, though, to get sex determination. So, again, we can only do this on our adult skeletons. Prior to this age, the skeleton is still growing, it's still developing, and many of the biologically based sex differences, which develop later in puberty due to hormones, are not yet present or are difficult to determine in our juvenile skeletons. So sexing typically relies on the two skeletal areas within the clearest sex-based differences, the cranium and the pelvis, right? We're going to be looking at the cranium and the pelvis really to help us determine if it's female or male. So, it takes two primary forms. Sex determination takes two primary forms, based on size, and sex determination based on shape. In general, there is a slight difference between modern human males and females, with males tending to be larger, right? They tend to be taller than females, so they have longer bones than females do. Males tend to be heavier and more muscular than females, so they tend to have more robust and more rugged bone features than females. However, there are individuals who do not follow that generalization, right? Taking into account our WNBA player versus our horse jockey. Furthermore, populations differ in their average skeletal size and amount of sexual dimorphism, right? So some populations are shorter than other populations, and that just has to do with the way that they have evolved for their environment sometimes. And we'll get there when we talk about human variation. We'll talk about why, certain populations might be more robust than other populations. Right. So sex determination. So how do we determine sex? Based on first the cranium, and then we will go into the pelvis. So the cranium is often used in sex determination because there can be pronounced differences in the sizes and shapes of male and female cranial features that can be examined quantitatively and qualitatively. So in general, cranial features and areas of muscle attachment are more pronounced and robust in males than in females. So males, and I'll tell you the differences right now, males tend to have a more pronounced or larger features. Things like the brow ridges, right? More larger features there. Larger areas of muscle attachments, so they have larger mastoid processes, right? So remember, mastoid process is that bump, that bony bump right behind the ear. So what I'm saying is in males, they tend to have a larger mastoid process than females do, right? So and you can go, if you live in a house and there are males and females in your houses, you can go around and try and feel that mastoid process and see if you do feel the size differences between the males and females. Similarly, the occipital region of the skull has more pronounced and rougher nuchal areas for the nuchal muscles in males than our females. The superior margin of the eye orbit in males is usually rounded and blunt, but it's sharper in our females. In females, the forehead tends to be more vertical and rounded with bossing or slight bulges. Males tend to have a broad, square-shaped mandible and chin, and females tend to have a rounder mandible and a pointed chin. The vertical side of the mandible, or ramus, rami plural, in males tends to be vertically oriented with flared gonial angles on the lower posterior of the rami, while female mandibles have rami that tend to slope backwards. Lastly, males tend to have slightly larger and more pronounced canine teeth, while females have larger and less pronounced canines. Right? So those are the cranial differences. And here we have it put together. I put this together for you guys to see the trait, and then the difference between males and females. So overall size, larger in males, smaller in females. Skull robusticity is more pronounced in males, less pronounced in females. Our brow ridges are larger and or thicker in males and smaller in females. Shape of the forehead, more sloped for males. Females, more vertical, rounded with bossing bulges. Occipital nuchal area of the skull tends to be rougher and more pronounced in males, right? Nuchal area, more nuchal muscle attachment in males. Makes sense that they would have more ridging. And then smoother and less pronounced in our females. So less muscle attachment, less ridging. Mastoid process is larger in our males. Again, more muscle or bigger muscles, more bone. So larger in our males and then smaller in females. Our superior margin of the eye orbits are rounded and blunt. So if I say that too fast and you're kind of confused, superior margin of eye orbits means the top of the eyeballs. Remember that. Males is more rounded and blunt, whereas it's sharper in females. Shape of the mandible or chin is more squared in males and more rounded and pointed in females. Your mandibular ramus is vertical with flared gonial angles in males, and then ramus slopes backwards in females. And then our canine teeth are larger, more pronounced in males, and smaller, less pronounced in females. And so this will make more sense, especially when we're looking at our primates and we look at sexual dimorphism, and we look at the amounts of male-male aggression in our chimpanzees, right? They have a high amount of male-male aggression, and therefore we can see in the male chimpanzees that they have these very large, very pointed canines, and they work very, very well for things like threat displays, where they flip the upper lip in order to threaten other males from approaching their territory or their women, essentially. They're female chimps. And so those canines are there for a very, very important reason. It's not because they're carnivorous, really. It's because they work as a threat display, right? So more male-male competition means more exaggerated sexual dimorphism. And I'm saying this now just so it kind of is implanted in your brain now, so that when we get to talking about primates in a couple chapters, then you will already start to see this. You'll pick up on this. Right? So that is sex determination based on traits of the cranium. Now we're going to look at the pelvis. So there are differences between male and female skeletons based on functional differences between the sexes. So the sexes have evolved to complement one another in sexual reproduction. Therefore, they have functional differences related to their different roles. Specifically, females can give birth. So the female bony pelvis must then accommodate the passing of a fetus during childbirth. Males cannot give birth, and then their pelvises cannot accommodate the passing of a fetus. So what do we see in the pelvis? What we see when we look more closely at the pelvis, we see a number of specific traits that vary between females and males, right? So I have here side by side, females versus males, right? What we see in one versus the other. And then I have another more cleaner chart for you guys in the next slide. So in females, the pubic bone is shorter supero-inferiorly, but rectangular or wider mediolaterally. So supero-inferiorly means top to bottom. So it is shorter top to bottom. And then mediolaterally means from side to side. So it is wider or more rectangular from side to side, right? In males, the pubic bone is longer supero-inferiorly, right, top to bottom, but triangular, narrow, mediolaterally, side to side. Our subpubic angle is wider in females, and it is more sharper and narrow in males. Our greater sciatic notch is narrower supero-inferiorly and deeper antero-posteriorly, right? So it resembles a boomerang shape. So antero-posteriorly is from front to back. So in our males, our greater sciatic notch is deeper supero-inferiorly and narrower antero-posteriorly. So it resembles a candy cane shape. So in females, it looks more like a boomerang. In males, a candy cane shape. And I have pictures of side-by-side pelvises so you can see the difference. In females, our pelvic opening is wider and heart-shaped. In males, it is more narrow. So the pelvic opening is narrow. And lastly, the coccyx. The coccyx in females is positioned more posteriorly, and the pelvic opening is less obstructed. So it's positioned more backwards, essentially. Not backwards, but pushed back. And then in males, the coccyx will curve anteriorly and partially obstruct the pelvic opening. That pelvic opening is the large hole in the center of the pelvis that is formed by the articulation of the two ossa coxae and the sacrum. The larger pelvic opening in females allows for the fetus to pass through the pelvis during childbirth, and passage through the birth canal is facilitated by the position of the coccyx in females. So we'll show you the chart, and then you will see the pictures. So again, pelvic trait, then males and females. Overall size and robusticity, larger, more pronounced in males, smaller, less pronounced in females. Pelvic opening is smaller and narrower in males. It's larger and wider and heart-shaped in females. Your coccyx is curved ventrally in our males, and it is curved dorsally in females. The pubic bone is longer supero-inferiorly, but triangular or narrower mediolaterally in males. And then it is shorter supero-inferiorly, but rectangular or wider mediolaterally in females. The subpubic angle is sharper and narrower in males, but wider in females. And then the greater sciatic notch is candy cane shaped in males, so it's deeper supero-inferiorly, and then narrower antero-posteriorly, and then boomerang shaped in females. So it is narrow supero-inferiorly, and then deeper antero-posteriorly. So to put it all together, so you have an idea of what I'm talking about, let's look at pictures. So again, overall size and robusticity, males tend to be larger and more pronounced than females are. So our pelvic opening is smaller and narrower in males, larger and wider and heart-shaped in females. The coccyx position, so again, pelvic opening is what I'm talking about here, right? It's more narrow here, more wider here. The coccyx position, again, more obstructed in males, right? It is curving ventrally towards the belly. And then in females, it is curved dorsally, so towards the back, right? And the coccyx, the pelvic opening is less obstructed. Coccyx position, again, I talked about that. Pubic bone, again, it is longer supero-inferiorly in males, so longer overall, and then narrower. And then in females, it's shorter overall and then wider overall. Subpubic angle in males is sharper, right? Narrower. In females, it is wider. And then the greater sciatic notch, which is what we're looking at here, right? Here. In males, it is more candy cane shape. In females, it is more boomerang shaped, right? So those are sex determination. It's pretty easy. With more practice, it gets easier to do over time. So after we've determined the sex of the skeletal remains, next we have to determine the age at death. And so after we separate out the individuals and we determine their biological steps, again, we determine their age. So it takes two primary forms. The first is based on developmental changes, and then the second is based on metamorphic changes. So we'll do developmental first and then metamorphic. So there are known rates at which the body grows and develops, and this is helpful when we are comparing a skeleton to the known developmental stages and determine then the likely age of that individual. These methods, these developmental change methods, are really suitable for aging our juvenile skeletons because their bodies are still undergoing these changes during growth and development. So a fully adult skeleton, however, cannot be aged then using these methods because they're no longer developing. So there are known rates of bone ossification. So this is process of bone mineral solidification and fusion that occurs as the body develops into an adult, right? Knowing the stages of these ossification processes provides us with a really useful aging method. So one example is the long bones of the body. They'll undergo particular ossification processes known as epiphyseal fusion. And we'll see that right now. So epiphyseal fusion is where the shaft of the bone, which is the diaphysis, and the ends of the bone, the epiphyses, fuse together with age. If a bone is recovered that is partially fused, that has partially fused epiphyses, the extent of fusion can help us determine how old this individual was. The more fused they are, the older they are, and the less fused, the younger. The epiphyses of the various long bones of the body fuse at different ages, with the clavicle being the last to fuse in the early 20s. And so here is a chart where it'll tell you, right, the ages at which these bones fuse. Right, our earliest being our humerus, and then our latest, it looks here like the sacrum, but also the clavicle as well, being the later one. Right, so these are ages of epiphyseal fusion in our in different skeletal remains. Right. Continue developmental changes. Right, there are areas where the bones come together and form sutures, and the extent of those suture closings can help determine age. So we're looking specifically here at the cranium. So cranial suture closing. In general, if the bones are separate, the individual is very young. So if the bones are fusing, but the sutures are very distinct, the individual's older. If the suture lines are less distinct or almost obliterated, then the individual is even older. So here with A, it's a juvenile skeleton. You can see here the fontanelles essentially are still open. So you have some fusing here among this suture line, but not up here. It's still the top fontanelle is still very much open. And then you have here they're fused, but not they're distinct. So it's older, but not too old. And then here they're pretty much blurred or obliterated. So it's an older skeletal remain. Right, so much closer look at the baby, right? I would assume they don't even have teeth, so I would say this cranium looks to be about 6 months old, 4 to 6 months old, right? Not too old, very, very young, right? Here again, a little bit older, and then almost obliterated, so a little bit older. Right. There are also, speaking of teeth, there are also known rates of tooth eruption. So this is what we call as teething. This is the time when the deciduous teeth are breaking the bone and the gum surface and they are growing in. So the deciduous teeth and the permanent teeth come in at different ages as the body develops. So the different teeth erupt at different ages and in particular order. So in general, the middle or the central incisors are first, and then the remaining teeth appear mostly from the front to the back of the mouth. The last teeth to come in are the last or third molars of our permanent dentition, which usually erupt in our late teens or early 20s. We call these teeth our molar teeth, or our wisdom teeth, because they come at a much later age than the other teeth do. Right, so our third molars in the back, essentially, those are our wisdom teeth. Again, they come at very specific intervals. So this is a chart that we use in order to determine given skeletal remains. So you're going to be, I think this is the exact x-ray you guys are going to be using for your lab, I believe. And then you are going to be looking at this chart to determine around where this individual is. We can see a second permanent molar is erupting with first permanent molar has already erupted. So we go through it and we figure out where along this this chart that individual is. And there is room for error, right? We can see plus or minuses here. So I have the age range at which this individual should be at, right? So those are developmental changes. We also have then age determination based on metamorphic or degenerative changes. So there are also these estimates. One of the primary areas to observe age-related metamorphic changes is on the pubic bone, right? These methods, these metamorphic or degenerative changes are suitable for aging adults as their skeleton and their joints begin to undergo degeneration over time. Changes occur specifically where we look at right here is along the pubic symphysis. So this is a joint where the two pelvic bones meet anteriorly along the midline. That can then be observed on the symphysial surface of the pubic bone. So many age determination methods have been developed from the morphological changes of the pubic symphysis. One of the most recent and the best aging methods is called the Suki-Brooks method. So in general, the prevalence of prominent and regular ridges on the symphysial face decreases as we age. The prevalence of rimming and boning nodules on the symphysial face will also increase as we age. And then the symphysial face becomes more concave as we age. So these changes occur at differing rates for males and females. So different ranges and charts are used to estimate the age at death of male and female individuals. So explaining it this way and looking at the picture might be very difficult. I do have linked in this week's module a video showing you specifically three pubic symphysis pieces, and then showing you what this means essentially. So I highly suggest you watch that before you do the exercise for this week, so you can really understand how to age based on these metamorphic or degenerative changes. So here are some examples, right? And then you'll see what phase they're in. Then you take that phase and then you determine male versus female, then how old that individual is. And it'll give you a description, right? So here's more close up for phase 1 through 2, 3, and then 4, 5, and 6. Right? We also have degenerative changes based in the teeth. So another method for aging adults is the analysis of dental wear. Over time, the chewing and the chewing surfaces of our teeth wear down from their continual use. So we can't tell an exact age based on dental wear. but it can tell us a relative age. And so in general, recent human populations have a fairly refined diet, and this results in very little tooth wear over time. As such, dental wear is not a precise aging method, like I said. In general, when determining the age of a skeleton, a more precise estimate can be obtained for juveniles than for adults. But this really gives us an idea, right? This is obviously not very worn down, so they're much younger, but if we see where the teeth are much more worn down, right, you're going to assume that they are older. Right, so here's an example, right? These teeth are very, very worn down. They belong to an older individual. So you can't even see the cusps on the teeth here. They're so worn down. Right? Next step is estimating stature, right? Once we have estimated age, we estimate stature. So a person's stature, their height is based in large part on the size of their bones. The person with longer bones will be taller overall. Someone with shorter bones will be shorter overall. This is useful in forensic cases because height is tracked in public records. So most researchers use measurements from the long bones. And so the long bones, particularly those of the arms and the legs, are used to estimate the stature because their length strongly correlates with their overall stature. So the anthropologists will measure the bone's maximum length, and they'll use an instrument that we call the osteometric board. But you can also use a tape measure. It's very easy to use. All right. This measurement then is entered into a formula that is dependent on the individual's sex and ancestry. So there are some, the results then is a stature estimate with a range or standard of error that gives an idea about how much error is possible in the measurements. So there are some issues to bear in mind with this estimation. Many of the formulas for stature estimation were developed using skeletons from a museum collection of people that died in the 1900s. So that's one issue. There have been gradual changes in body size and height among populations in the last century, and it is unclear if the skeletal populations in the last century, the skeletal measurements are accurate for more recent forensic cases. So it's not 100% accurate, but it gives us an estimate. And then the formulas for the tibia are probably not as precise as the formulas for other bones. Right, so here's a bone in an osteometric board. We take the measurement and then we plug it into the formula. Here are the formulas. Again, it depends on the bone that we're looking at, and then as well the descent of the individual. Right, I believe in the exercise it's a male of European descent that you're looking at. It will tell you. So that's estimating stature. Next, we identify pathology. So aside from identifying the remains, bioarchaeologists and forensic anthropologists are also interested in any indicator on an individual skeleton that is not related to normal variation but is related to disease, pathology, or metabolic stress. So they must also distinguish between anti-mortem pathology and perimortem pathology. So anti-mortem is any pathology that developed at any point prior to the individual's death. Perimortem is pathology or trauma that developed around the time of an individual's death that may have contributed to the death. This is important, right? Perimortem pathology is important to forensic anthropologists because they can then be asked to comment on the manner of a victim's death in legal proceedings. So the manner of death includes circumstances surrounding the death that may have contributed to death, such as traumatic injuries. However, the forensic anthropologist does not determine official cause of death. Right? The official cause of death is a medical determination made by a medical examiner or a coroner, often in conjunction with information from the forensic anthropologist. Remember, our job as forensic anthropologists is to create the profile. We create the profile to help the medical examiner, the coroner, and they determined the cause of death. Right? To properly identify possible contributing factors to the victim's death, the forensic anthropologist must be able to distinguish pathology that occurred earlier in life and did not contribute to death. So, anti-mortem pathology can tell us valuable information about how an individual lived and what their overall health was like during life. So, for bioarchaeologists, anti-mortem pathology tells us about the health of one person, and pathology indicators in many individuals in a population can help build a picture of the overall stress and lifestyle in the past. For forensic anthropologists, we're looking at anti-mortem pathology to aid in the overall identification of a victim, right? So say you have a bone break and you can determine that this happened around when the the individual was around 7 years old. You can then look at medical records and see, or public records and see if there's any instances of that, right? That's just an example. There are several categories of anti-mortem pathology. This includes pathology related to overall health and metabolic stress. Often, when the body is under nutritional stress, it compensates by slowing down bone growth and focusing on metabolic energy elsewhere. When the nutritional stress is over, the bone then begins growing regularly again. Stress indicators are routinely used in the analysis of skeletal remains. So we're looking at teeth, right? Teeth. If an individual has a period of nutritional stress due to lack of food or severe illness, when the teeth are growing during childhood, the enamel will stop forming on the tooth crowns for some time. When that period of stress is over, has passed, the enamel forming process will resume where it ordinarily should be, not where it left off. The result of this then is a gap or horizontal line with reduced enamel called a dental enamel hypoplasia. Because we have specific knowledge of when teeth and enamel are formed, we can then use the location of dental enamel hypoplasias to obtain an accurate estimate of the age at which the nutritional or illness related stress occurred. Right, so those are nutritional related stress. Overall health, metabolic stress, pathology associated with nutrition, specifically disease, and then chronic conditions. Okay. Another nutrition related pathology is the presence of dental caries. We also call them cavities. These are areas of the teeth that have undergone demineralization and decay due to acid exposure. The acid is formed naturally by oral bacteria as the mouth begins to break down food to start digestion. If acids are produced in higher quantities due to what we eat or not properly cleaning away through proper dental hygiene, caries can form. The presence of dental caries is much higher in populations with the agricultural lifestyle than in our populations with a more foraging lifestyle. This is because our agricultural lifestyle is often based on a diet high in carbohydrates, which then results in more acid in the mouth. More acid in the mouth means more dental caries or more cavities. In addition to pathology associated with nutrition, there is pathology associated with more specifically with disease. So we can look at things like iron deficiency anemia, and this is a condition caused by a severe lack of iron in the body, often due to a disease or parasite in the body, potentially due to dietary stress. The primary indicator of this is a type of bone destruction called Protocorotic Hyperostosis. This is usually seen on the cranial bones. So what happens is that the marrow cavity will expand. It causes the smooth cortex of the flat bone in the cranium to look porous and spongy. And I'll show you a picture of that shortly. And then aside from nutritional and disease-related pathology, there are also pathologies associated with chronic conditions. So this is like osteoarthritis, which is common among humans. And this is where in humans, it's a condition where accumulated wear and tear on the joints results in the loss of their cartilage linings. A loss of these linings causes changes, both bone formation and bone loss in the underlying bone. Without healthy cartilage as a cushion between the bones, bone surfaces at a joint end end up contacting each other directly. What happens here is it can result in eburnation, which is the bone gets polished and shiny due to repeated direct contact between the bones at the joint, or it can cause the formation of spicules on the bone, or what we call osteophytes. And this happens around the margin of the joint surface. Osteoarthritis is common in highly mobile joints such as the hip, knee, and lower back. So just to give you an idea, a picture of all of this, we have dental enamel hypoplasia. So you can see the horizontal lines here, where they were undergoing stress and it caused that dental enamel hypoplasia. Here we have dental caries, so cavities, right? This is Protocorotic Hyperostosis, right? So instead of being smooth, we have a very spongy looking texture, right? So this is all Anti-Mortem Pathology. Then we have Perimortem Pathology. And so as with our Anti-Mortem Pathology, there are different types of Perimortem Pathology. Perimortem pathology is very important, especially in our forensic context. So most perimortem pathology is distinguishable from antemortem because it involves severe traumatic injuries. So knife wounds, blunt force trauma, gunshot wounds, etc. I'm going to explain the differences between all of these. So knife wounds, how do we know we're looking at a knife wound? Knife wounds are often indicated by large cuts or nicks in the bone, right? This is when we get a hard blade making contact with bone tissue. It leaves behind small cut marks or indentations. The size, the length, and the depth and the shape of the cut marks can indicate the size and type of blade used, and the orientation of the cut marks can indicate the angle of the thrusts. Blunt force trauma. This is trauma that results from coming in contact with a blunt object. It can occur through falling, through being hit with an object. And so blunt force trauma is indicated by the presence of a depression in bone at the point of impact. There can also be small bone cracks or fractures that radiate out from the depressed area due to the force of the impact. Gunshot wounds or any projectile trauma can be very obvious in the skeleton. If an individual has been shot, there may be clear points of entry and exit, which is indicated by holes in the bone. The entry hole is smaller and has more discrete edges than the exit wound, which is larger and has an inward sloping beveled edge. The size of the entry wound can be used to determine the maximum possible bullet size, and the orientation of the entry and exit wounds can be used to indicate the trajectory of the shot. Right, so entry wound can give us the size, the maximum size of the bullet. Exit wound can help us determine the trajectory of the shot. Some traumas leave very distinct damage to particular bones. So something like strangulation requires a lot of force to be applied to the throat, and in the throat you have a small fragile bone known as the hyoid bone. It is likely to acquire damage as the result of this force. So therefore, if you have an unhealed fractured hyoid bone, it's usually indicative of some kind of choking or strangulation. Often we experience traumatic injuries that don't contribute to death. These must be distinguished from our other injuries to understand the manner of death. So a fracture must be distinguished from any injuries that may have contributed to death. The distinction is based on whether the bone has healed through remodeling. And we'll get an example later on at the end of the semester of a specimen that has partially healed fractures. We'll talk about it a little bit. If the fracture was treated well, it will heal very cleanly and will leave little evidence. If the fracture was not treated well, then the bone may heal incorrectly and the two sides might not line up together or correctly, or they may be extra bony formation in the area of the fracture. Right? So let me give you some photos to look at. This is actually going with our antemortem. This is osteophytes or spicules of bone. So this is osteoarthritis that we're looking at. But if we're looking at perimortem, here is blunt force trauma, right? We can see, again, it is a depression and you have radiating fractures going outwards. Then you have here a bone break, right? This bone break was not properly taken care of. Therefore, the two ends of the bone don't align very well. If I ask you to look at this bone and to tell me what bone it is, you should be able to tell me what it is, right? Even looking at it. Give you a second to guess. So if you guess that it is the tibia, you are correct. This is the tibia bone. So this bone is the tibia. It is the left tibia. I had to determine left and right on my hand really quick. It is the left tibia. Right. Right, so the last thing that we look at, the last forensic anthro step we do is ancestry. No, the first step. Sorry, now we've done the bioarchaeology and forensic. The first additional step is looking at ancestry. So forensic anthropologists are asked to determine a victim's likely ancestry because this information can aid in the attempts to identify an unknown victim or victims. Bioarchaeologists may be interested in determining ancestry in bioarchaeological populations to help study past migrations or population movements, familial relationships, and other popular trends. But really, this is used more for forensics. There are two groups or types of traits that can be used to recognize morphological indicators of ancestry, so our metric and our non-metric traits. Metric traits are osteometric quantitative traits that are measurable on the skeleton, such as the length or breadth of certain bones. Typically, dozens of measurements of different skeletal areas are taken, and these measurements are incorporated into complex statistical analyses. The use of metric traits is based on the principle that people in different regions will have different sizes, proportions, or degrees of robusticity, something that we'll talk about in the next lab. And our non-metric traits, these are anthroposcopic qualitative traits that are not measurable. So instead, they're traits that somebody either has or doesn't have, right? They're scored as either present or absent and can be found on the cranial and postcranial skeletons. So different ancestral groups are more likely to have different combinations or suites of traits. So one example is shovel-shaped incisors as a non-metric trait. So it's a non-metric trait and refers to the unique incisor shape in which the lingual side surface of the tooth, or the tongue side, has distinct indentations that make it look like the spade of a shovel. This tooth shape, and it's one that I actually have, you'll feel it if you take your tongue and you feel the back of your incisors and you go from bottom upwards and you feel a ridge, right? Or like you feel these ridges here. It means you have shovel-shaped incisors, right? And you'll feel this bump up here too, right? This is a tooth shape that is most commonly found in people with Asian or Native American ancestry. Another non-metric trait is the Malar Tubercle. This is a bony projection that extends from the inferior edge of the zygomatic bone. This tubercle is pronounced in people of Asian ancestry, but it's usually absent in people of European and African ancestry. In addition to these traits, anthropologists may also look for non-metric variations in the shapes of skeletal features. So we can look at the nasal root, the superior nasal margin. It's low and rounded in our African populations. It is high and peaked in European populations, and then low and peaked in Asian populations. So anthropologists can study suites of traits like these to try and get a person's possible ancestry. It's important to emphasize that there is individual and within population variation in both non-metric and metric traits. So within an ancestral group, not everyone will have the exact same measurements or all the same non-metric traits. Further traits that may indicate biological ancestry do not inform us about the cultural or ethnic affiliation of a person during life. Finally, we are dealing with the ranges of different populations and how they overlap. Right, so this is an example of shovel-shaped incisors. Here we have the nasal root that we're looking at, so between the three populations that we talked about. Right, so that's the first step, the first extra step. And then the second extra step, and the last thing we're going to talk about, is calculating postmortem interval. So the forensic anthropologist is also usually asked to estimate the postmortem interval for a victim. Right, this interval refers to the time that has elapsed since death. Generally, as more time elapses, it becomes more difficult to pinpoint the exact time of death. During the early stages of decomposition, there are regular phases of insect activity. So if insects are present on the bone, they can be used to determine postmortem interval. Similarly, soft tissue decays fairly quickly and at known rates. So the degree of soft tissue decay can also be used to estimate time of death as well. If enough time has passed that there is no longer soft tissue or insect activity on the body, determining the postmortem interval becomes much more challenging. Bones decay too gradually to be used alone for calculating elapsed time intervals, so calculating the postmortem interval is further complicated by variations in the environment and decompositional conditions, right? So again, depending on the area, the temperature, right, it can lead to changes in calculating this postmortem interval. All right, so that is it for this chapter. Right, the next chapter I believe we are looking at is human variation, so we're going to get into a little bit more about variations in the skeletal system, a few of those, as well as overall body robusticity. Okay.